Looks like the bulk of our guests are here, so thank you all. I love that we are a timely crew. It is a pleasure to meet all of you guys. If I have not met you in person, my name is Renee Croto, and I am the Executive Director of the Northern California region for the Leukemia and Lymphoma Society. And I'm going to go off script for just 1 second. Don't worry, Annie, but if you guys were reading the comments as we were joining on, I just thought it was so beautifully said what Chris mentioned, that what a great way to recognize Blood Cancer Awareness Month by coming together for this virtual lab tour. And you guys are in for a treat. So thank you, thank you. And again, this is just an incredible evening. For those of you guys are used to LLS, we like to do things in person, and obviously this is a little bit different. We are first in the nation to attempt a virtual lab tour, and we are so excited for what you guys have in store tonight. You know, just be patient with us. We have mastered virtual events after 6 months, but occasionally there are tech glitches. And so if that is the case, we just ask that you be patient with us, but we have practiced and we ensure that today's event will go smoothly. Really quickly, I'm going to transition just to some housekeeping. You all probably are quite familiar with Zoom by now, but if you didn't realize, you are on mute right now, and that's intended just to cut down background noise. You're welcome to ask questions throughout the event using the chat feature, but at the end of tonight's program, we will open up for Q&A for you to connect with our speakers. And if anytime tonight you have any technical issues, you can private chat LLS staff, and one of our incredibly talented staff members is on the other line and will navigate with you to make sure that you can maximize the experience today. And one last thing I wanted to note in our housekeeping is that this event is being recorded, which is important that we notify all of you guys and to be compliant with California law. So with that, I just want to thank each of you guys so much for being here. We are so fortunate to have such committed partners with the Leukemia and Lymphoma Society that support us during these trying times of a pandemic, smoky skies, and so much more. And you know, if we think about this though, through this all, cancer isn't canceled. Our world has been turned upside down. But every year, the fact remains true that 10 times the number of people who have died of the COVID pandemic in America die of cancer. And so this statistic doesn't change, and we need your guys' support to do that. And today you're going to hear about a lot of the resources and what is happening behind the scenes so that we can find a cure for blood cancer. So we thought we'd start off tonight's program with something that's slightly interactive. And so I want to encourage you guys to pull up your chat box on the bottom right of your screen and put in there what you would describe if you could imagine a world without cancer. What would that look like? So please take a moment to type that in, and I'm going to pull up the chat myself and read through some of these responses so we know who is on the line and what that would look like. All right, things are coming in right now. So it looks like more survivors. Gwen says she could retire. Peace, I love that. Coffee with sisters, yeah, more time with family. Those are great ones. Happiness, no worries. Barry, so perfect. You'll hear from him in a little bit. It would mean that my family would still be with us. Yes, that is the case for me, as I have something very personal. Oh, these are great. I'd still have time with my parents, and my brother wouldn't be battling right now. Yes, I'm sorry to hear about your brother. Families celebrating life rather than death. That is what we are here for, Stephen perfectly said, and healthy, happy people. So thank you all for sharing, and continue to put your remarks in there. As you think during this program, that is what we are aspiring for. That is why we are here. I know I've shared with many of you that I lost my father to lymphoma 10 years ago. And I think about the idea of having Christmas with my daughters and they get to know their Grandpa Jeff. And that's so important and keeps me going. And so these thoughts are really should be our driver and inspire us. So thank you all. I'm gonna give a couple brief updates, but I know that you guys are here to hear our incredible speakers. So I wanna share just briefly with you some LLS updates, and then both at a national level and at a local level. And then I'll hand it over to our first speaker. So first, as you can see here on the screen, what's incredible is that the Leukemia and Lymphoma Society has helped 55 of the 65 FDA approvals since 2017. That is an incredible statistics. We are investing in smart science and smart research that is coming to fruition with therapies and getting us one step closer to a cure. And this is just the beginning. And we are working tirelessly to fill treatment gaps and make more promising research to become fruition. So one example of what we're doing, and I hope that Gwen on our call will be able to speak a little bit more about this, but it's our new children's initiative. And this program is revolutionizing pediatric care. Therapies for children have not changed in 40 years. Just this week, I was meeting with a donor whose son is 22 now, but went through treatment when he was 15 years ago. And I thought to myself as I was meeting with him, he had the same treatment 15 years ago for his son as someone diagnosed today. We need to change that, and LLS is committed to that. Our work is to turn the needle in developing new treatments that are less harmful and toxic so that children can live long, healthy lives. And I know we all can get behind that. So LLS, as you can see on the slide, is committed to raising $25 million and investing in childhood cancer research over the next 5 years. And I'm so excited for the updates to come and what the progress that we're gonna make because of this investment. And then on a local level, right here in Northern California, we are so fortunate to have two phenomenal medical institutions that are making a change. We're gonna hear from Stanford today, but as you can see on the slide, since 2018, $8.5 million has been invested by the Leukemia and Lymphoma Society to UCSF and Stanford, and their research has targeted AML, various types of lymphoma, and multiple myeloma. And I know, for many of you guys on the line who work hard to raise funds and are donors and give so generously to your mission, you can speak with confidence and boldness that your dollars are making an impact locally, but then so much broader, as you're gonna hear from Mark today, of the long-term impact that we're gonna have. So on the slide are our featured speakers and what tonight's program is going to look like. First, we're gonna start off with brothers Stephen and Jeff Hamill, who will open up with a mission moment. And then we are thrilled to have our Chief Medical Officer, Dr. Gwen Nichols, talk about the impact of COVID-19 is having on cancer research. And then lastly, our keynote, which we're all so excited, and I can't thank him enough for the hard work of how to approach this in a new, innovative way. Mark Smith is gonna show us and take us with live updates that he filmed throughout his lab and throughout the campus of Stanford, and then share what's happening with some of his cutting edge research that's happening. So on that note, I'm going to introduce you to two very special people, Stephen and Jeff Hamill. Yes, they're brothers, and they have a very personal connection to the Leukemia and Lymphoma Society. Stephen is a longtime standing donor for LLS, and Jeff is a non-Hodgkin's lymphoma survivor of 13 years. He received care both here at Stanford and the City of Hope. Steve and Jeff, thank you so much for joining us today. And on that note, please take it away.
Thanks, Renee. That was an impressive and compelling introduction to the evening. Our engagement with LLS and Stanford was inspired by Jeff's lymphoma diagnosis. Jeff will share his story. He lived it, and he has a great short story to share. By way of introduction, our family has a history of cancer. Our family has been ravaged by cancer, from brain to prostate to esophageal to melanoma to lung cancer. In fact, I and my three brothers are all at this point in our lives cancer survivors. We have a protocol in our family when faced with life-threatening cancer to seek out centers of excellence. And when Jeff was diagnosed, top of mind was Stanford and LLS as some of the best, if not the best, in the field of addressing the issues that Jeff was facing, the threat to his life. Since Jeff's diagnosis, our family has become actively engaged with LLS, and LLS has become a family priority in our charitable giving to a number of cancer research organizations and other philanthropic pursuits, but again, top of mind for us. We give because we simply want to accomplish three things. We want to improve survival rates. We want to improve the patient experience. And when you're faced with a diagnosis of cancer, we don't want that to be the end of the sentence. We want a but. We have a remedy, we have a treatment, and a potential cure. What's important to us in an organization that we support is a laser focus on improving survival rates, accelerating the time from research to patient, proof, and moving the needle on the road to a cure. LLS research is a compelling charitable contribution for us because LLS has a philosophy, as does Stanford, that we've got to get better each day and improve cancer outcomes. It's not accepting what is, but what could be in the future. And in our experience, supporting a number of cancer organizations, LLS stands alone in its long history of progressive treatment and cures. And as evidenced by Renee's comment on 55 of the 60 some odd treatments were LLS supported. I'm glad to be here tonight and join each of you in learning more about LLS and Stanford's research as a center of excellence. I think tonight will just be further evidence of the need to continue our support of this valuable and important work. And with that general introduction to our family and our priorities, I'd like to turn it over to my brother, Jeff, who lived the diagnosis, and he has a compelling story to tell. Jeff?
Thank you so much. And I'd like to thank everybody that's on the Zoom or the call, whatever you want to call it, tonight. I really appreciate it. When I was first diagnosed in 2007, I made a commitment to myself that I would always answer the call and certainly reach out and help anybody afflicted with this. I've had that opportunity several times over the years, and it's great tonight to be able to do this. Again, I was diagnosed in 2007. I was living in China at the time, working for Starbucks. And I went home for my annual physical, and my doctor called me and said that my blood work was unusual. And he asked me to come back to the States. And when I got back, he surprised me with a bone marrow test. And I was diagnosed with stage 4 non-Hodgkin lymphoma. I learned my diagnosis when I got back to China because I didn't think I had anything wrong with me. I just went back, and I immediately was kind of blown away, but I called my brother, my mentor, my confidant, my friend, Steve, and he said, we're going to get you back to the States as soon as possible, and we're going to get you into a program where we can get a second opinion. And that's when we went to Stanford. This is all happening in a pretty quick order. When I got to Stanford, the doctor immediately said, you do have non-Hodgkin's lymphoma, but there's more to the story. And I'll never forget that. My spleen was about 8 times the size of a normal spleen. And the diagnosis was splenic marginal zone non-Hodgkin's lymphoma, which is about 1% of the cases of non-Hodgkin's lymphoma. The suggested immediate action was splenectomy. I lived in Southern California, or my main home was in Southern California. So after that diagnosis, I went to City of Hope in Duarte, and then I was in surgery within 2 weeks. because the doctor immediately agreed 100% with the Stanford diagnosis, which was exceptional, detailed, and I was able to have my spleen removed, which in a pretty short order, within about 3 months, we were able to determine that I was in remission. With that, I was able, to stay with City of Hope for about 15 years. I had one flare up over that time and had Rituxan treatment in 2010. But since that time, I have not been able to be in remission, virtually remission, for the rest of the time. So I'm very lucky, I'm very blessed. And importantly, I think my engagement with Stanford, helped by Steve, I will say saved my life, because I was working in China, like I said, and had I just continued on kind of dealing with it slowly, I think that I would have probably had a problem with my spleen. So that's kind of, the big part of my story is that. And I just would like to say I've seen progress in treatments at City of Hope and otherwise with this disease. And LLS and the work and the donations have greatly impacted people like me. And I'm grateful to LLS. I'm grateful to Stanford and City of Hope as well. And my story is just for me. I want to do everything I can to help, and that's why I'm here tonight. And I thank you very much for the opportunity to do this.
Thanks, Jeff, and good evening, everyone, and add my thanks to all of you for being here tonight. My name is Barry Cheskin, and I'm the board chair of the LLS Board of Directors for the Silicon Valley Monterey Bay region. My wife and I also have a strong mission connection to LLS. Over 20 years ago, I lost my dad, who was just 56, to non-Hodgkin lymphoma. Just this past year, Lisa lost her dad, and I lost my father-in-law to CLL, chronic lymphocytic leukemia. And on a positive note, thankfully, we have seen our nephew Nichols successfully treated at Stanford for acute lymphocytic leukemia. He's doing well. He was diagnosed at 13, and he's now a sophomore at Cal Poly. My wife and I have been volunteers and strong supporters of LLS for many years. And we support LLS because of the mission connection that I just described. But also we support LLS because it gets stuff done. And as you heard Renee talk about, it's an impressive organization, and its impact and its ability to influence and support the treatment of blood cancers is really extraordinary. And that is a perfect segue to our next speaker. I am honored to introduce Dr. Gwen Nichols. Dr. Nichols is LLS's Chief Medical Officer. She oversees all of LLS's scientific research portfolio, patient services, policy, advocacy initiatives. As a physician and a scientific researcher, she's dedicated her career to advancing cures for cancers and has done a remarkable job. Thank you for being with us tonight, Gwen.
Well, thank you so much, Barry. And I really would like to thank the Hamill brothers. I think that was just lovely. And your sibling love is so evident even by Zoom. So I really, it's very touching to see. So thank you for sharing. It really, really warms my heart. I am really excited to be here. I'm actually not in the Bay Area now. I am joining you from New York, and where the air is better, and but we're equally all in the same virtual world. And I think it's important to talk about how COVID-19 and the impact that has had on our research is so important for our supporters to understand. So you've you've heard about, we're rightfully proud, I think, at LLS of the fact that our research has supported impactful programs and impactful researchers like Dr. Smith that you'll hear from later on. The 55 of 65, and it's always changing every couple of days, there's something new, is something to be rightfully proud of. But I have to tell you, I think while we all know what COVID has done for people and their work and their jobs and all of us, and financial impacts, and obviously the horrible loss of life, I don't think everyone has understood what it has done to scientific research. So as you can imagine, if you think about it, laboratories really had to stop research in March. Laboratories are not good places for social distancing. I know when I had a lab at Columbia in New York, we had like 400 square feet with 4 people in it, and we thought it was a luxury. There's no way to socially distance in that kind of environment. And in fact, many of the laboratories that are in hospitals, people were sent home because the hospitals had to focus completely on treating patients and not put any of the staff at risk. And so while we all know the research is essential, that wasn't considered an essential working. This has continued well into the summer until we were able, and this is around the world. This is not just the United States. And as you know, LLS supports research all over the world, the best, wherever it comes from. All the labs came to an abrupt halt. Cell lines had to be frozen away. Animals had to be left and not, they were tended to, but the experiments couldn't continue. Much was lost, but more than that, a big gap in time where the type of bench research couldn't be done. But researchers, and I think hopefully Dr. Smith will tell you, did not go home and feel sorry for themselves. They really have taken up the mantle to figure out what kind of work they can do remotely. What kind of work can they do that involves looking at data, analyzing data, sharing things in a virtual world. It's only now that labs are starting to get back, and many of our researchers have described that their staff is going back in shifts because they don't have the kind of space and ability to keep people socially distanced appropriately. So fortunately, many of the people in labs are young and they like to work in the middle of the night. And then the rest of the staff works the daylight hours and they talk to each other virtually. But it had, I chuckle about this, but the truth is it's a big gap, and it's a gap that can be really impact the speed of research. I wonder if you can put up the slide that I had. I thought this might be interesting for people because COVID had, and the way that drug manufacturers have banded together and the FDA to speed vaccine development along is really extraordinary. If you look at the red boxes at the bottom, and I like this train picture because it helps me talk about a number of different things. There are lots of stops along the way to those 65 new treatments that we, 55 of which we helped develop. That isn't just done in a couple of years. It's long time, consistent funding of really quality research, including basic research and drug discovery, which for a new molecule often can take between 3 and 6 years. That's followed by preclinical testing, which LLS also supports, testing to create the drug in a format that actually can be given to patients. Is it effective? Who is it effective for? Clinical trials also take quite a long time to be done in a proper manner and as carefully as possible, often 5 to 7 years, followed by a couple more years of FDA review before a new therapy can get to a patient. So we're talking on average around 12 years from the time we support the first research until the drug, if it's successful, is available for patients. COVID has derailed this process. It derailed clinical trials. It delayed research. I believe that we have the will to get this back on track, but we need support for that. The other reason we need support, I'm not as worried about the research of the big scientists. They're well supported. Their institutions support them. They can continue their work. What I'm really worried about is younger researchers, folks who have kids in school and mortgages and med school loans and all the things that other young people have, but they also have to apply for grants every year. And LLS, along with many, many other not-for-profits, in COVID had to switch all of our in-person fundraising. And we are, left with fewer dollars this year than we normally would have for research. This means for young researchers, grants are going to be harder and harder to get. If it was only LLS, that would be fine, but it's not. It's across the board, cuts, decreases, delays in research funding. And some young people are going to just say, I can't do this anymore. This isn't the profession for me. And they will leave academic research. And I think when you hear from Dr. Smith about how important academic research is and why LLS is supporting these researchers, to the process of bringing new therapies to patients, you'll understand why I'm so concerned about a generation of young researchers potentially being lost, because it's just too hard to stay in the game. We are really working very, very hard to find funding and find ways to help particularly these younger and middle career scientists, along with obviously all the support we are giving to more senior scientists. We need this COVID time not to impact early career scientists. So due to all of this, your support is really more urgent than ever. And I encourage you to help us be instrumental in changing this and keeping the discovery train on the track. I think that support for your local institutions, wonderful institutions like Stanford and UCSF, is really, really important. Your local LLS team can tell you all about that. Both institutions will be part of the LLS Children's Initiative, both through funding pediatric research, but also through a novel clinical trial that we will be bringing to fruition in calendar 21 that will help translate the discoveries into new, less toxic therapies for patients with an international collaboration. So thank you for the opportunity to talk to all of you. And I really look forward to hearing and seeing Dr. Smith. So I will hand it back over to Barry, but thank you so much for giving me the opportunity to speak with you this evening.
Thanks, Gwen, for that great update. I know I and we all always appreciate hearing from you. I am really excited to introduce our next speaker, who's worked very hard to, you will see, bring the lab to you virtually this evening. Dr. Mark Smith of Stanford Medicine. He is head of the Medicinal Chemistry Knowledge Center. He has previously received an LS research grant for AML research and is an outstanding longtime volunteer as well. Mark has participated on the Scientific Grant Review Board for the LLS's Translational Research Program to help accelerate bench to bedside research. He works in Stanford's Drug Discovery Lab to ensure the most promising research becomes the next cure. Thank you, Mark, for being with us today and for all the hard work you've put in over the years and for bringing the lab to our supporters tonight. Mark?
Well, thank you very much. So I'm not sure if many people have actually been to Stanford, so we're going to start off the presentation with a quick tour around Stanford's campus, because this is where you would actually come if you were here and coming to visit my lab in person. So it's kind of famous for those who've been here before is the actual quad. Stanford was founded in 1891. The first graduating class was in 1895. Actually, one of the graduates was Herbert Hoover, who became obviously the 31st president of the United States. When he actually left Stanford in 1944, he handed his house over to the university, and that's now the residence for the current president of Stanford University. It's a very beautiful campus. Interestingly, we have a lot of art on campus, including the biggest collection of Rodins outside of Paris. So I highly recommend you check out the Gates of Hell at the Cantor Art Center. It's also a famous building. It's the Stanford Church, which was actually designed by Jane Stanford. Stanford was constructed with sandstone, which was quarried just south of San Jose, but it turns out sandstone is not a very robust material for buildings that you hope would last for hundreds of years. This day, all the stones actually quarried from a place just north of Paris. Stanford actually owns a quarry there. So I found that quite fascinating. And when you look closely to these buildings, you'll see fossils in the limestone that's on this. This is our CCSR building. It's actually where the lymphoma clinic is. Professor Ron Levy is downstairs. He's the person who was behind the drug Rituxan that Leukemia Lymphoma Society funded many years ago. Across the way from the CCSR building is our stem cell research building. Here, Irv Weissman, who's a pioneer in the area of leukemia research, who just actually did the checkpoint inhibitor CD47 that Gilead scientists purchased a few weeks ago. Across the way from the stem cell, we have the Beckman Center. It's a famous building in terms of just the sheer volume of Nobel Prize winners in chemistry and medicine who've actually operated there. Now we walk down Discovery Way. This is actually an art exhibit at the university in the medical school. If you look over to the left of the screen, you'll see a lot of artwork on these stones, which really basically communicate 150 years of medical discoveries, of which a lot of they highlight a lot of the ones which were conducted actually at Stanford. We come now into one of the newer buildings. This is the Clark Center and home of Stanford's BioX Institute. This is where really scientists from across multiple disciplines are looking to understand and improve their knowledge of biological systems inside the body. And the architecture of this building was an inspiration to the building I currently work in, which is one of the newest at Stanford, which is the Stanford ChemH Institute that we'll go over to shortly. What's important at Stanford in the medical school is that we bring scientists with very, very different backgrounds to get to know one another, because we have a knack of speaking very, very different languages. And that was the inspiration behind this new ChemH Institute. As you can see here, this is actually our building. That building there, the kind of circular building, is our pub. They decided it would be a good way to bring scientists on campus was to build a pub on the medical school side. Going into our courtyard, we've got a Neuroscience Institute on the left and ChemH on the right-hand side. Now, ChemH stands for Chemistry, Engineering, and Medicine for Human Health. And the focus is really to bring chemists into the laboratories of physician scientists and biologists and kind of educate a new wave of students who are fluent in both biology and chemistry, because chemistry is fundamentally important in drug discovery. We look at disease on a molecular level and actually to design molecules to inhibit a lot of that work has to be done by chemists. And usually at universities, the chemistry department, the biochemistry, the biology departments, and the medical schools are all isolated. So they wanted an institute to bring all those kinds of scientists together. And as you can see just from the interior of our building, it was designed with collaboration in mind. The idea here is that people can get out the labs and talk to scientists from other laboratories. So it's quite unique. This is actually my laboratory. We have our own lounge where we can have meetings with our collaborators. It's a fairly large lab. We have over 3,000 square foot of space. It's the kind of go-to laboratory for ChemH. We're in a premier real estate, I should say, for the new building. I was lucky enough to get involved with the design of the building. This first part is our structural biology section, where we manufacture, purify, and crystallize proteins to help with structure determination. We're walking through here. This is actually an equipment room. This is a fifth-year graduate student, Jackie Carozzo. She researches immuno-oncology. She's actually very focused on activating the innate immune system, and the potential application of her research is not just blood cancers, but actually all cancers. And it's a very exciting area, and I'm going to talk a bit more about Jackie's pioneering research. She's coming to the end of her PhD. She should graduate maybe in May and probably go on to do a postdoc, but we've just been publishing a lot of the work that she's done lately. Now she's in the tissue culture room. She's just, I think, treating with ethanol her gloves so that she can actually run some cell assays. Here she's just prepared in some buffer, and I think on this particular day she was about to run a drug that I actually designed against a line of triple negative breast cancer, just to see what the activity would be and the efficacy in that process. This is me doing some molecular modeling. This is how I design a lot of the drugs that the group works on. It's very sophisticated software. We managed to get an X-ray structure of a protein co-crystallized, and from that information I was able to design a therapeutic. What I want to do is design a small molecule like the one here in yellow, which binds really, really tightly to the protein of interest. That's the basic concept of small molecule drug discovery. And that's why chemistry is so important to the whole drug discovery process. Now we're back in my lab. You can see the offices that my students work in there to the left. We have some storage rooms, cold rooms, and now we're heading over to where we do our purification work. This is state-of-the-art equipment that many academic labs do not possess in the chemistry world. This is what one would expect to see at a Pfizer or a Novartis company. This is called a flash chromatography instrument, and the postdoc there is just purifying some material. Those instruments cost about $35,000 each. Now we're in the standard synthetic chemistry part of the laboratory. Synthetic chemistry, it's kind of like cooking, but it's got a very dangerous edge to the work that we're doing. This particular reaction a postdoc's performing has a tendency to set on fire or explode. In the past, I know one of our colleagues' labs here at Stanford did set one on fire with that very reagent that the postdoc just put into that reaction mixture. So we have to be incredibly safe. It's a tough environment. You have to wear a lot of protective equipment because of the risks associated with the work. But this is how all general synthetic organic chemistry is performed here at Stanford. So I'm fortunate. I probably have the best setup that I'm aware of in academia. In fact, I think I've got the biggest fume cupboards, the best facilities. Like I said, we only opened this laboratory in January of this year, and then we shut down soon afterwards for a period of time. This is my microwave synthesizer. It enables our group to do 60 reactions overnight. It really is about efficient heating, and we can do it under high pressure and high temperatures that one can't produce under normal atmospheric conditions. That instrument alone cost me about $60,000. Now we're in the mass spectrometry part of the lab. Very sophisticated equipment. This is actually what weighs our molecules. We want to know each molecule, the exact mass of it, to make sure that we've made the right material. Now someone is actually preparing a sample for nuclear magnetic resonance spectroscopy. This is a very important method of analytical chemistry that's vital to our work. We'll put the sample, we're actually using an isotope of hydrogen in that particular sample, and then we put it in this very, very large magnet, and this will determine the exact structure of a molecule, where the atom's located there. This, though, technology is the premise of magnetic resonance imaging technology that one has when they have cancer. So my own personal story, going back with the Leukemia Lymphoma Society, goes back to 2005 when I was diagnosed with a rare form of B-cell lymphoma. Since then, for every year, I've been working out with the Leukemia Lymphoma Society team in training program to do some fundraising to give back to other patients less fortunate than myself. However, I've actually been through 11 relapses during that period. Most recently, I was in treatment in March of this year, just before the shutdown. I like to think I'm very fortunate and very healthy. This was a picture taken when I was working in NYU. I used to work in the pharmaceutical industry out in New Jersey. I was taking Rituxan treatment, and ironically, now I'm friends with Ron Levy, who developed the treatment. But 1 hour after that photograph was actually taken, I had a severe immune allergic response to the treatment, and I was lucky enough that a friend was there to set off the alarm and get the crash team in to save my life. So that kind of really inspired me that maybe I should leave the pharmaceutical industry, go back to academia, and really help develop a new kind of, well, personally have an impact on drugs of the future in cancer. That's important to me, and not just blood cancer, although that's a very strong personal connection, but for all cancers. I've met a lot of patients over the years suffering from cancer, and the bottom line is it kind of sucks. No one likes cancer, and I think we need to get away with superior treatments going forward. So when I arrived at Stanford, having left Big Pharma and returning to the academic environment, I really wanted to get involved with a project in the blood cancer space. And I immediately connected with Professor Kathy Sakamoto. At the time, she was the head of pediatric hematology oncology at Stanford. And she was telling me how desperate they are for new treatments for pediatric AML. You know, it is the deadliest form of leukemia for children. And as adults, though, it's even worse, and the chances of survival remain pretty poor. Now, Kathy had identified a protein called CREB, cyclic response element, cyclic AMP response element binding protein. Is this transcriptional protein here, and it's actually binding to a piece of DNA. And she found that it's been upregulated in a number of AML cases, and if it's upregulated, your prognosis is actually quite poor. So she wanted to target with a small molecule, a way to inhibit this actual process from occurring, in the hope that we would alleviate and maybe result in cures in combination with other therapies. So she asked if I could come in and design a molecule to do that process. We reached out to the Leukemia Lymphoma Society to get funding through their Hit to Lead program. This provided us with $200,000 of much needed research funds. It enabled me to bring in a medicinal chemist to work under my supervision, to actually do synthesis in the lab, to flush out some what we call structure activity relationships, and provide her with molecules that could actually result in efficacy in an animal model. We were very successful in this process. Interestingly, though, with the structure activity relationships we developed, we stumbled across an old molecule which was designed to treat tapeworm many years ago, and we realized this was similar to the structures we'd been working on. So we took this molecule and we tested it out, and it showed great efficacy, resulting in a 50% cure rate in AML mice that were given patient-derived samples of AML. And that has recently received additional funding from the LLS to go to a proof of concept study in a phase 1 clinical trial. This would never have happened if it wasn't for our donors at the Leukemia Lymphoma Society being able to put the money into the LLS to actually distribute. We couldn't get the funding by traditional mechanisms such as the NIH. So for me, I really saw the impact of the value that the LLS brings to our research. Given the success on that project, that's when the LLS asked if I could actually review their grants as well as part of their translational research program, and I've been doing that for the past, say, 4 or 5 years. Another project I was excited to share with you today is the work of my colleague Lingying Li and our graduate student Jackie Carozzo, who you just saw in the video. Lingying came to me when she joined Stanford. She did a postdoc at Harvard, and she said that she was looking at innate immune activation, and she had kind of discovered that there was this this molecule here. It's what we call a dinucleotide. It's 2 pieces of RNA joined together in an unusual manner. And it turns out that this is a molecule that cancer cells give out as a danger signal that they're in trouble, that they've mutated, and they send it out in order to recruit the immune system to the tumor microenvironment to enable destruction of the cell. At the same time, cancers decided that it doesn't want this signal going out. So it's developed a method to destroy the signal. So it silences this innate immune, as we call it, an immunotransmitter. So Lingying reached out to me and asked if I could actually design a drug to stop cancer from blocking this signal from going out. And it didn't take too long before we had some very, very good success on this particular program, when we were able to stop and silence the mechanism that cancer uses to eliminate this particular signal. We've been using this actually primarily for triple-negative breast cancer, but it should work with lymphoma. The reason why I'm particularly interested in it is because I've always been very, very sensitive to radiation therapy. I get my radiation therapy, ironically, at Stanford, some colleagues with my oncologists. And I respond really well, but I've always been curious to what the mechanism was. And now we've got a very thorough understanding on a chemical level of what's going on when one receives radiation therapy. And I think we'll see a future where we could actually dose people with lower radiation. But the other important thing is this is going to actually activate what we call cold tumors. A lot of the current immunotherapy treatments, they use the adaptive immune system, but they're completely ineffective with many cancers because there's no infiltration of the immune system in the tumor microenvironment. This methodology is going to enable T cells to penetrate the tumor microenvironment. We already have evidence for this. We published it earlier this year in Nature and Cell, and a small biotech company has actually recently licensed this work, and they're hoping to take the drug that we've developed into phase 1. I'm hoping next year, but we'll see how COVID's actually affected the progress of that. As you've heard, COVID's had a major impact on our research this year. My lab was shut down for 3 weeks. I got a special exemption. We do shift work. My group come in at 6 o'clock, half the group come in at 6 o'clock in the morning. They work through to 2 o'clock in the afternoon. Then the next group come in and they work from 2:30 all the way through to midnight if they can. They're usually itching to get as much work as they can because we've really restricted the hours. They get tested for COVID once a week. And we have all these protocols in place that people have to follow. So not only do I do research in oncology, but I do a lot of research in infectious disease and have a couple of programs for looking at inhibitors for coronavirus as well as other potential pandemic viral infections. Because of that, I kind of wanted to share a little bit of information and the correlation that people aren't aware of between viruses and cancer. There's actually a number of viruses, and some of you may be aware of this, that cause cancer. An example would be hepatitis C, another one hepatitis B. They both cause liver cancer. Human papillomavirus is obviously famous for causing cervical cancer, but there's also a link between some of the herpes viruses and B-cell lymphoma as well. Back in the 60s, people were looking at nucleic acids as potential treatments for viruses and also cancer. And I kind of wanted to use this cartoon, if anything, to highlight the similarity in the biological mechanisms at play. So what's happened here is a virus has entered the cell, it's uncoded, and it's left this squiggly kind of figure here that is a polymerase protein. That's the representation. Now the genetic material here coming through of the RNA of the virus is being copied. We've now got a template. That template's going to come back in and it's going to manufacture more viral proteins. So all these different colors, they represent a nucleic acid like C, G, T, and A. So as more viral protein, viral genetic information is being manufactured, more viral proteins will be made and more viral particles will be made. This will result in an immune response on the innate immune system, the stuff that I've been working on actually for cancer. It would also solicit the T cells to come fight the virus. In the case with coronavirus, it's probably a too aggressive a response to the virus pathogen, and thus one gets quite sick. Now, these are the actual building blocks for our genetic information, this specifically being for the RNA building blocks, the difference being that they have a hydroxy group at the 2 position. And the technology to inhibit this process is actually quite simple. We just make a structural derivative of these nucleic acid building blocks as shown here. And what happens is when we give these drugs to the patient, they will actually they will act as a Trojan horse. The virus that will think that it's actually a natural substrate. In this instance, maybe it thinks it's a cytidine, but it actually isn't. It's like a Trojan horse. It gets in there and it stops more genetic information being transferred. Now, mechanistically, this is what we've used very successfully for a number of viral infections like hepatitis C, HIV, herpes virus, amongst others. But it actually is the underlying backbone for a lot of chemotherapeutic treatments that have been developed over the years, specifically for leukemia. In fact, one could argue that the technology that was helped us develop AZT for HIV really stemmed from research that was being funded for leukemia back in the 1950s and 1960s. Mechanistically, these drugs all work the same. The difference is that ones that we actually use for virology are designed very selectively for the viral protein, and the ones that are used for cancer, like cytarabine and chloraphrobine and gemcitabine down here, they're designed to inhibit the genetic information of for a human polymerase, and thus it stops the rapidly dividing cells from processing, and they actually kill the cells. So they're quite toxic in that respect. But I thought it was interesting for the audience to understand that funding research in one area can actually have a dramatic impact on research in another disease area. And the historical investment in cancer research has really helped us understand mechanisms and find new treatments for viruses. Just in closing, this is my team. It's a little bit dated. I've got 3 new team members since they're past and present, but these are the superstars who do all the work in my lab, and they're the ones developing the new technologies as we're going forward. They're the future leaders, scientific leaders, I like to think. And with that, I'd just like to thank you all for your continued support for the Leukemia and Lymphoma Society. Obviously, for me, it's very, very personal, but I really appreciate all the efforts that everyone have done. And all I can say is that I'm not a fan of mediocre research. And when it comes to, you know, for me, it's a great honor to be on the scientific review board of the LLS. And I can just personally guarantee to you, I will not allow the LLS to fund mediocre research. I only expect the best research to be funded going forward. And that's what I expect out of every single member of my research lab. We aim high. Mediocrity is of no interest to me. We don't have enough time to waste on mediocrity. And I'll be happy to take any questions in the Q&A session. Thank you.
I am seeing the comments come in and just want to reiterate what everyone is saying there, Mark, and it's thank you. That was incredible and just an amazing way in this environment to bring the lab to us. And I know because you've sat on many rehearsals with us, and I know how much time you spent capturing that video for us, and we just can't say thank you enough for that. I do want to open the floor up to Q&A. If anyone has any questions for Dr. Smith, Dr. Nichols, or any of our other speakers, please feel free to chat it into the chat box. While those questions are coming in, I am going to ask Dr. Gwen if you wouldn't mind speaking a little bit about the Children's Initiative, because I know that's something you are very passionate about, and I love to hear you speak on it.
Well, sure. Thank you, Renee. And you may have noticed it in the chat, but I was so happy you mentioned Dr. Sakamoto because she's a friend of mine from college. So I'm so excited to see that she has had such success and has worked with someone as extraordinary as you, Dr. Smith. So the Children's Initiative, one of the roadblocks, and I think I'm going to talk mainly about the end result of what we want the Children's Initiative to come to. That's new therapies for children. And I think Don Proctor has said a few things in the chat as well about the blood cancers being relatively rare diseases. We're 40% of the FDA approvals, but only 10% of the cancer market. So they are relatively rare. And if you divide up the many different subtypes of lymphoma, for instance, this makes each cancer even rarer. And so the childhood cancer takes that to a different level in terms of rare disease. And so the impetus for pharmaceutical developers to focus on childhood cancer and childhood cancer biology just isn't there from a business point of view. Now, it's very easy to say, oh, well, pharma's bad, but this is a business, and many of you may be business people. You'll know you're going to choose a larger market if you have the market. So what can we do to make it more cost efficient, faster, and help pharma to make it operationally possible for them to develop therapies that are specific for children? Well, one of the things we need to do is focus on childhood biology, because the truth is that children are not just miniature-sized adults, and AML is a perfect example, and I'm sure Dr. Sakamoto would be the first one to tell you, childhood cancer biology overlaps with adult cancer biology, but if you look at AML, maybe a third of the abnormalities that drive the cancer are overlapping. 2/3 are not, but no one is developing drugs for that other 2/3. They're only developing drugs for the larger adult market and then testing them in children. What we can help make happen is make sure that every child gets sequenced and gets their biology examined so that we know what the differences are and even what the drugs we need, and we can go to Dr. Smith and say, we need this, something that targets this specific thing for, this specific protein, this specific signaling molecule, this specific pathway, so that we can have drugs for kids. But that isn't the simple answer. You also need enough patients to actually test it and know if it's effective. And the only way to do that is to do that in collaboration. So we are planning an international clinical trial for children whose disease has relapsed after standard therapy. And this will include Europe, Australia, Canada, the US. We have agreement from the Children's Oncology Group in the US and 5 different AML consortia in Europe. And we already have Canada and Australia on board. So this is a very exciting way to share data and to have pharma see us and say, okay, you're going to be checking the sequencing and the biomarkers on every child, so we can choose to develop the drugs in the patients that you have in this large collaboration that have the abnormalities are drug targets. And it puts the child in the center rather than the drug in the center. And I think that that's the very exciting path that we're pushing forward with the Children's Initiative. So as you can hear, I could talk about this all night, but we have a lot more, I'm sure, questions and we want to get to Dr. Smith, but thanks for the opportunity, Renee.
Absolutely. And yes, as you just said, you are so passionate, which is why I wanted to ask you to speak on that. In the meantime, we have had a few questions come in. Our program did run a little long, and so for those of you that 6:30 need to step out, we respect that, but we are going to continue with a couple more questions. And so I'm going to start here. Mark, this one is for you, and it said, you mentioned that research findings are often benefit other diseases. How do researchers go about sharing such findings with other labs and researchers?
It's actually something that was very, very important at Stanford, was to actually create environments where people talk to one another. Usually medical schools are quite distant on campuses to the biology labs, the chemistry labs, and so on and so forth. The Stanford Hospital is about 300 yards from my window here. I have physician scientists in my building. I found what was so powerful about coming back to academia is I can talk to a physician scientist whenever I want on whatever the disease area. And we had our physician scientists in the same buildings, but you know, hematology, oncology will be one floor, gastroenterology will be above that, cardiovascular research next door. People talk to one another, they become friends from other labs. So that knowledge is shared. And this was actually part of the design of the buildings. How do we get these people into the same environment so they talk to one another and do become friends, and then they can solve real world challenges? In my group meetings, I actually have MD, PhD students show up. I've got synthetic organic chemists showing up, biologists, bioengineers. They get to become friends. And when someone has a question medically related, we've got the MD, PhD student who's just finished medical school to turn to for some advice. I'm actually a central figure as well, because I see so much. I probably see more small molecule research going on at Stanford than anyone else. And because I run the Innovative Medical Accelerator at Stanford as well, where people write their proposals that I have to review each year, I can actually link projects and people together. I recently done that when I actually put Professor Kathy Sakamoto in collaboration with an endocrinologist. And it just turned out I saw some synergy with the research they were doing, that they could talk to one another. They've never spoken before. And they started sharing some compounds for different indications, but they wouldn't have known about it if we didn't have this kind of setup. And I think this setup's fairly unique to Stanford. Like I say, many schools do not have medical centers co-located with everyone else. It helps me out because I learn more about medical problems. As a chemist, you don't know what struggles doctors face. And when they come to me and they say, yeah, this isn't ideal, we use this drug Temo for glioblastomas, and it's so unstable, it's not that efficacious, and you start to think about it, I'm like, oh, we can re-engineer your molecule to make it better. How would you like to dose it to a patient? As a simple thing like that, they never think about, but we can think about it. So now what we're doing is deploying chemists throughout campus to start solving these really molecular engineering problems that shouldn't really be problems if a chemist knew about them. But we don't know what the medical problems are. So that was what's important, and that's why the medicinal chemistry knowledge center actually exists. The director of ChemH, Professor Chetan Costa, realized that there was this massive knowledge gap in academia, and they called medicinal chemistry the valley of death, where good projects go to die, good biological projects go to die. And without medicinal chemistry support, which is often only found in the pharmaceutical industry, because traditionally it's taught in the pharmaceutical industry. So I spent a long time in big pharma working for Roche Pharmaceuticals, both in Palo Alto and then in Nutley, New Jersey. And I actually decided to start my own biotech company. And that's when Stanford said, hey, could you come in and teach faculty how to do this? And I also run the undergraduate entrepreneurship program. So we teach our undergraduate students to become biotech entrepreneurs. We just kicked off this program. We give 4 teams of 3 students on just address an unmet medical need that you're passionate about. We train them up, we bring in industry experts, and we move them forward. So we really are trying to change the culture of drug discovery on campus. And it's not one initiative, it's multiple initiatives.
There's so many things I want to respond to, but for sake of time, I'm going to move on to the next question. And this is going to be our last question for the night. So thank you all. So the question is there research being done to design better personalized protocols that take individual genetic composition into account to improve outcomes? And examples listed were Hispanic versus Caucasian versus Indian.
You might want me to take that one,
Yeah.
Dr. Smith. Yes.
Thank you.
So there, absolutely. And I think there's more than just, so there's a tie-in to the work that Dr. Smith is doing, and that is that there are genetic differences in drug metabolism based on SNPs and all kinds of other, that's the simplest explanation, changes in variations, not mutations, but variations that have to do, that are oftentimes passed down genetically, or that skew into certain ethnic, racial, and racial groups, and can make a huge difference in both response to therapy, but also incidence of cancer. And we're finding out so much more about that. But personalized protocols need more people to participate in the clinical trial process. And so part of the problem is that only 5% of cancer patients ever participate in a clinical trial. And if we want all these new discoveries and these new molecules to actually make it to the clinic, we need to make it easier for more people, and more people of color, of different ethnic backgrounds, of different social backgrounds. Even where you live, you all have the benefit of being at, in the epicenter of, wonderful science and wonderful clinical trials, but rural patients don't have that. And so we need to do a lot to get more people participating so that we can personalize and understand the differences that we see scientifically, but can't test because the people aren't participating in the trials.
Well, thank you. And thank you everyone who attended tonight. This does conclude our program. And I just want to say from the bottom of my heart, just how much we are so grateful to partner with each of you. I hope that you found tonight, as we brought the lab to your home, that you walked away with one little nugget of just the incredible work that we are doing at the Leukemia and Lymphoma Society. As you can see, your support is more critical now than ever. And Gwen did a great job showcasing that, of just how we've been impacted with young investigators. That really has resonated with me as I've learned that. If you'd like to learn anything more about the Children's Initiative, about some of the stuff that Dr. Smith spoke about, please just let us know and we will connect you and continue the conversation. And lastly, just as we sign off, a sincere and genuine thank you to Dr. Nichols, Dr. Smith, phenomenal program. Thank you for going above and beyond. And also to Stephen and Jeff for sharing your journey with LLS with us. So with that, have a lovely evening, everybody. Thank you so much, and hope that our paths can cross in person one of these days. Have a great night. Bye-bye.